Across the globe, 81% of school-aged youth fail to meet baseline physical activity guidelines. The biological inputs required to maintain cellular homeostasis are disappearing. In clinical settings, medical professionals frequently prescribe physical activity to adolescents, primarily as a metabolic countermeasure, a tool to manage BMI and burn calories. That view misses the underlying biology. Exercise operates as a pleiotropic biological signal. It acts on multiple systems simultaneously to shape insulin sensitivity, construct bone architecture, and drive neuroplasticity. In developmental biology, the DOHaD framework, the Developmental Origins of Health and Disease, establishes fetal life as the primary window where environmental inputs permanently program adult physiology. The decade of adolescence, driven by the hormonal cascade of puberty, represents the second great window of developmental plasticity. Unlike fetal programming, this adolescent window is highly accessible. It can be modified through behavioral interventions in real time. As puberty begins, rising frequencies of gonadotropin-releasing hormone pulses force the brain and body to undergo massive structural and functional reorganization. Multiple systems are reprogrammed at once. The hypothalamic pituitary gonadal axis matures, the skeleton achieves peak mass, metabolic pathways shift for rapid growth, and the prefrontal cortex densely myelinates. The human genome expects these transitions to occur in an environment requiring continuous, moderate physical exertion. Today, adolescents navigate this developmental phase in an environment of ultra-processed foods and extreme sedentary behavior. Sedentary behavior during the adolescent decade deprives the body of the exact epigenetic signals required to successfully navigate pubertal maturation. The pubertal transition is triggered by a massive hormonal surge. Estradiol, progesterone, growth hormone, and insulin-like growth factor 1, or IGF-1, flood the system. Growth hormone and IGF-1 specifically drive a state of transient physiological insulin resistance. The body intentionally reduces its sensitivity to insulin to redirect glucose toward rapidly growing tissues. This physiological insulin resistance is 25 to 50% more pronounced than typical adult insulin resistance. Historically, continuous moderate physical activity provided the mechanical and metabolic stress necessary to buffer this intense metabolic shift. Modern adolescence presents a double mismatch. There is a biologically driven slowdown in movement during puberty, which is now compounded by highly sedentary screen time replacing incidental physical activity. When the physical exercise signal is removed, physiological insulin resistance does not resolve after puberty. It persists and progresses. This progression directly maps to the early onset of pathological states, driving visceral adiposity, accelerating pubertal tempo toward early menarche, and increasing the risk of polycystic ovary syndrome and metabolic syndrome. Without the mechanical and metabolic stress of exercise, the adolescent body lacks the biological software required to safely metabolize the hormonal fuel of puberty. The mechanistic bridge linking adolescent physical activity to lifelong disease prevention operates at the genomic level through epigenetic reprogramming. In this context, epigenetics refers to movement-based inputs actively stripping chemical tags, known as methyl groups, from key gene promoters. This changes which genes are active without altering the underlying DNA sequence. Definitive proof of this mechanism emerged in 2014 from researchers at Sweden's Karolinska Institute. They designed a highly controlled study where healthy individuals performed one-leg cycling. Subjects pedaled with only one leg for 45 minutes, 4 times a week, over 3 months. The scientists took muscle biopsies from both legs before and after the 3-month intervention to track genomic changes at the cellular level. The results revealed that over 5,000 sites on the genome of the muscle cells had developed entirely new methylation patterns. These specific epigenetic changes appeared strictly in the exercised leg. The resting leg, sharing the exact same systemic environment and blood flow, showed no such alterations.
The mechanical stress of exercise directly hypomethylates the promoter for PPARGC1A, upregulating PGC1-alpha, the master regulator of mitochondrial biogenesis. This forces the cell to construct new mitochondria, increasing oxidative capacity, while permanently upregulating GLUT4 proteins to expand insulin-stimulated glucose disposal. Exercise actively edits the genome's metabolic software, creating a highly localized permanent metabolic phenotype that persists long after the movement stops. This same programming logic extends from the muscle to the skeleton, where developmental windows are finite and cannot be reopened once puberty concludes. The adolescent skeletal window operates primarily during Tanner stages 2 and 3. In this brief peripubertal phase, bone is exceptionally responsive to mechanical loading, and 90% of adult bone mineral density is achieved. This chart plots bone mass accrual. Girls beginning impact exercise before menarche accrue twice the bone mineral content of those starting post-pubertally. This peripubertal bone capital is locked in, permanently dictating fracture risk in an 80-year-old body. Simultaneously, the adolescent brain undergoes its own structural reorganization, heavily refining the prefrontal cortex and the limbic system. The central neurobiological driver for this rewiring is brain-derived neurotrophic factor, or BDNF, secreted in massive quantities during aerobic exercise. Similar to the muscle biopsy data, exercise induces DNA demethylation at BDNF promoter 4, upregulating expression and drastically increasing synaptic plasticity. Clinically, this rewiring translates into persistent improvements in reward sensitivity, executive function, and emotional regulation. This plasticity is especially relevant for the vulnerability of the luteal phase of the menstrual cycle. Conditions like premenstrual dysphoric disorder, or PMDD, are driven by a paradoxical brain sensitivity to the normal luteal neurosteroid allopregnanolone. During puberty, the adolescent brain is highly susceptible to stress-induced distortions in GABA-A receptor composition, which creates this exact neurosteroid sensitivity. Exercise-induced BDNF directly regulates GABA-A receptor expression and trafficking, structurally buffering the brain against this luteal phase sensitivity. Furthermore, exercise normalizes HPA axis tone, counteracting the cortisol hyperreactivity that amplifies neurosteroid vulnerability. Adolescent exercise structurally sculpts both the physical skeleton to prevent future frailty and the neurobiological architecture required for lifelong emotional resilience. The act of physical exertion programs the genome, the skeleton, and the brain simultaneously during the adolescent window. This profound biological programming extends far beyond the individual, actively projecting physiological resilience deep into the next generation. Under the DOHaD framework, emerging evidence establishes that maternal exercise habits long before conception program offspring metabolism through epigenetic mechanisms in the germline. The timing is critical. Establishing robust exercise habits during adolescence occurs before oocyte maturation cycles begin in earnest. This creates a highly favorable epigenomic and mitochondrial environment within the oocyte that persists for decades before any conception actually occurs. Breakthrough 2025 data demonstrates that maternal exercise specifically improves offspring endurance, alters muscle fiber composition, and enhances overall metabolic phenotype. This flowchart illustrates the precise intergenerational transmission pathway. Maternal exercise upregulates placental SOD3, which activates fetal liver AMPK. This sequentially triggers TET1 and TET2 enzymes, catalyzing active DNA demethylation in both the F1 and F2 generations. The clinical result for the offspring is increased hepatic expression of metabolic genes, yielding improved glucose tolerance, enhanced metabolic flexibility, and a measurably reduced risk of obesity and type 2 diabetes. This specific epigenetic pathway actively interrupts the intergenerational transmission of metabolic risk that currently drives the epidemic of cardiometabolic conditions. Adolescent exercise prescriptions function as intergenerational medicine, architecting the biological resilience of both the woman and her future children.